investigation. This study is related to one of the topics, which is drug delivery problem. Why drug delivery is considered as a more cheapest alternative of producing, and you know that consuming and expensive process imported for targeted delivery, for target, because some drugs, you know, can be toxic. Sorry, it was some problem. Can you hear? Can you hear
Yes,
me?
we can hear you, but it looks like there is poor internet connection, and now
I'm sorry. What about now? Is it
now
okay?
it's much better. It's much
Yeah,
better, and
I'm
we can
sorry.
see
Yeah.
the presentation.
I'm connecting to you from Kharkiv, and it's possible to have some problems with internet. Sorry for this. So I continue that drug delivery and targeted drug transportation is one of the most important thing for anti-cancer drugs, which can be toxic or can be low soluble in water. And that is why in recent years, some constructions, some nanostructures were proposed for drug delivery of anti-cancer drugs. You can see it's in the slide. It can be liposomes, nanosomes, micelles, dendritic, some other nanostructures. Among of these structures, non-organic nanoparticles, and in particular molybdenum disulfide, attract very high attention of researchers, because these nanoparticles can provide not just drug delivery and targeted transportation of drugs, but also can be considered as a purposal transportation platforms, because this material is very effective for photothermal therapy. For example, molybdenum disulfide, you can see the structure of this compound. They can use for drug delivery, and it's already used in medicine for today. Not just for transportation of drug, but also for photothermal therapy disease also, because this material can effectively absorb laser waves and produce thermal effects, which can provide synergetic effects in anti-cancer therapy. From the biophysical and nanobiophysical point of view, we are interested in such side of this problem as intermolecular interaction between the drugs molecules and nanoparticles which are used for drug delivery. And we, during such investigation, we are trying to answer on some questions. For example, whether the drug delivery systems can be effectively uploaded with the drugs molecules, and whether the intermolecular interaction of these medicines with nanoparticles do not affect the structure of the drugs, and whether these drugs can survive and can provide their therapeutic activity. And related to this side of the drug delivery problem, we formulated the aim of the current study as to characterize physicochemical properties of nanobiohybrids of molybdenum disulfide nanosheets with some selected anti-cancer drugs, and to evaluate nanocomposites applicability prospects for drug delivery based on the results of combined experimental and theoretical modeling nanobiophysical study. As for the drugs which we selected for our study, you can see the structures of these compounds which are presented in this slide, we have taken the widely used anti-cancer drugs like doxorubicin, it's A letter, two CO derivatives of purine nucleobases, it's ceapurine B and ceoadenine, and also widely used anti-cancer drug 5-fluorouracil, it's D letter. So all these drugs are used for today in therapy, and most of all, for example, these derivatives of nuclear bases are low soluble in water, but for doxorubicin, which is effectively used also without any drug delivery systems, such molybdenum disulfide can be used, and we know this from the literature, for providing this synergetic effect, which I already mentioned, which is photo and thermotherapy provided by molybdenum disulfide nanoparticles. Our experimental method of choice was laser desorption ionization mass spectrometry technique. Marina Vadimovna already said some words about this technique. We prepared our nanocomposite molybdenum disulfide nanoparticles with anti-cancer drugs by ultrasound treatment, and also dried the samples, and they were positioned on the metal plate. And after this, using the laser radiation, it was laser desorptionization of the samples, and we analyzed the spectra obtained. I should mention that for the experiments we used a Center for Collective Use of National Academy of Science of Ukraine, which is situated in Institute for Surface Chemistry in Kiev. For a theoretical modeling, we used the ab initio DFT method, which give us possibility to model complexes, we called their bio-nano hybrids of molybdenum disulfide nanosheets with the molecules of anti-cancer drugs, which I mentioned. It was doxorubicin, thiopurine, thiadenine, and 5-fluorouracil. For modeling of nanoparticles, we took nanosheets containing 27 atoms of molybdenum and 54 atoms of sulfur. First of all, I will show you the experimental results, because for us it's important to have experimental basis, which is a basis for following theoretical modeling. At this slide, I would like to present to you the LDE mass spectrum results for investigation of bio-nanocomposite of molybdenum disulfide with doxorubicin. You see the spectrum of pure doxorubicin and the spectrum of nanocomposite of molybdenum disulfide with doxorubicin. And you can see that comparing all the spectra, you see that in the spectrum of nanocomposite, we can see all the characteristic peaks which are characteristic for doxorubicin, including the molecular ion of doxorubicin, which is cationized molecule of this drug. I should say that doxorubicin is, that widely used drugs, and we have already in mass spectrometry libraries the spectra of this compound. All these peaks are already identified before us, and they are related to active adducts of doxorubicin, and which is important that all these adducts contain this part of doxorubicin molecules. It's a system of 4 cycles. anthracyclinous part of these molecules, which is very important for biological activity of doxorubicin and anti-cancer activity in particular. So comparing this spectra, we can say that we see in the spectra of nanocomposite of molybdenum disulfide with doxorubicin all characteristic adducts of doxorubicin. So we do not see any changes in chemical structures for doxorubicin based on our LD mass spectrometry data. I just would like to show that why it's this part, anthracyclinic part, is important. Preserving of this part is important for doxorubicin because the major theory of anti-cancer activity is the intercalation of this part of this anti-cancer drug molecule between the into the DNA molecule of fast growing and fast dividing cancer cells. At the next step, when we analyze the nanocomposite, we used theoretical modeling to model the possible intermolecular interaction of doxorubicin molecule with molybdenum disulfide nanosheets. And we used two possible initial geometries to model the possible interaction. The first one is position of doxorubicin molecule under the plate of molybdenum disulfide nanosheet, and calculation and modeling of such structures. At the finally, we have obtained the result that doxorubicin can form stable stacking complexes with molybdenum disulfide nanosheets with the geometry which is presented at in Fig 2. In two projections, it's top projection and side projection, and you can see that really it's stacking complex where the plate of these four anthracyclinic cycles of doxorubicin molecules interacting with the plate of molybdenum disulfide nanoparticles. And the energy of such interaction is quite big. It's you can see the calculated energy, intermolecular energy in such complexes. It's more than minus 50 kilocalorie per mole. And the second initial geometry which we modeled is interaction of doxorubicin with the edges of nanoparticle, molybdenum disulfide nanoparticles. I should say that in the literature, there are data that catalytic activity of molybdenum disulfide, and I should say that molybdenum disulfide is characterized by high catalytic activity. So this catalytic activity from the literature data is related to activity of these edges atom of molybdenum disulfide nanoparticles, or with the places of some defects in these nanoparticles surface defects. So when we selected as an initial geometry side interaction of doxorubicin molecule with molybdenum disulfide nanoparticles, at the end of modeling, at the end of calculation, we obtained the structures of covalently bonded doxorubicin molecules with molybdenum disulfide nanoparticles. And this Fig 3 show two possible complexes, covalently bonded complexes, which show the stable hybrids, bionanohybrids of doxorubicin with molybdenum disulfide nanoparticles. In the first complexes with the energy minus 89 kilocalorie per mole, we see that this complex is supported by three covalent bonds between oxygen atoms of doxorubicin and molybdenum atoms of nanoparticles. The second stable geometry is characterized by more energy, even by more strong energy. It's about minus 137, and it's supported by four covalent bonds, but these bonds already formed between C atoms of carbon atoms of doxorubicin and molybdenum atoms of nanoparticles. So we can see that two types, non-covalently and covalently bonded complexes, can be formed, but we should say that because nanoparticles of molybdenum disulfide and other two-dimensional dehalogenides of metals are characterized by high surface in comparison with the perimeter of edges, we should say that we consider that the amount of molecules interaction by non-covalent bonds are bigger in comparison with the molecules which can form the covalent bonds interacting with the edges of the nanoparticles. In contrast to the situation with doxorubicin, for which we did not observe any spectral changes in molecular structure of doxorubicin, some certain features of LD mass spectra for nanocomposite of molybdenum disulfide with thiopurine and thioadenine were observed. And we should say that we predicted such changes and such observation because for CO containing biological active drugs, it's known that in the presence of molybdenum disulfide, which is catalytic active, as I already said, it's possible to observe the oxidation processes of these sulfur-containing compounds, and such processes can be related to the formation of dimers. The reaction, the scheme of reaction is presented at this slide. And this dimer forming with the creation of SS covalent bridges between the molecules. And actually, we have seen the confirmation of our prediction analyzing the LD mass spectrometry data for nanocomposites. At this slide, for example, we presented the spectra of nanocomposite molybdenum disulfide with thioadenine. It's also these systems were measured in a positive and negative ion mode. And also here you can see the comparison between spectra of poor drug, poor thioadenine, and nanocomposites of thioadenine with molybdenum disulfide. These are positive in red, positive ion mass spectra. In green, we show negative ion mass spectra. If we can compare the spectra of poor drugs and the spectra of nanocomposite, we can see that in the spectra of nanocomposite, the relative intensity, I will try to show by cursor, the relative intensity of peaks of covalently bonded dimers of thioadenine in comparison with the intensity of molecular peak of thioadenine is growing in the nanocomposite. And we can see the similar picture in the positive ion mass spectra as well as in negative ion mass spectra, because in negative ion mass spectrum for poor thiopurine, we almost do not see the peak of the dimer. Based on this spectral data, we can conclude that we can see the intensification of oxidation process and with the formation of this dimer for thioadenine in presence of molybdenum disulfide within the nanocomposite. And The same results we observed for the nanocomposite of molybdenum disulfide with thiopurine. And also you see the spectrum of the relative intensity of spectrum of dimer of thiopurine in nanocomposite is growing here in positive ion spectra and here in negative ion spectra in comparison with the peak of molecule of thiopurine. But for system of molybdenum disulfide with thiopurine, we have found also very interesting effect because in the spectrum, in the positive ion mass spectrum of nanocomposite, we have found the peaks which were not observed in the spectra of pure thiopurine. For example, peaks with the MZ 137, and 159. And we from the literature we could identify, based on the literature, we could identify these peaks as molecular ions, protonated and cationized ions of another derivative of purine nuclear bases, hypoxanthine. And also which is very important and very impressive, that the biggest peak in the spectrum of nanocomposite is the peak, the most intensive peaks, it's the peak of also covalent dimer of this hypoxanthine. So based on this spectral data, we can conclude that CR derivative of purine nuclear bases in the presence of molybdenum disulfide can show the chemical transformation. For example, covalent dimerization or formation of hypoxanthine. It's also presented in the scheme here. And it's very important for anti-cancer activity of this drug because exactly existence of sulfur atom is very important for providing anti-cancer activity of thiopurine as well as thioadenine. So based on this spectral data, we can say that these molecules can process and which can be result in chemical transformation of these drugs. As for modeling of thioadenine and thiopurine interaction with molybdenum disulfide, we should say that this, first of all, for these molecules, we will found the most stable tautomers, because these molecules are characterized by quite big amount of tautomers. And at this slide, you can see the most stable tautomer for thiopurine. These two types of tautomer, sion form and seol form. For thioadenine, the most stable tautomers both are seol, but the first one is N1 and the next one is N3, seol tail form. These two most stable tautomers were used to model the complexes of thioadenine and thiopurine with molybdenum disulfide nanoparticles. And at this slide, you can see already the final results of this modeling of this calculation with the energies of interaction between the components of the complexes, which are mentioned also under the pictures. You can see that for thiopurine, it's keta tautomer, it's enol tautomer of thiopurine interacting with molybdenum disulfide nanoparticles. We can see that also similarly to the situation with doxorubicin, thiopurine can form two types of stable complexes with nanoparticles. It's non-covalent complexes in which the plate of thiopurine molecules in interaction by stacking interaction with nanoparticles, and energy of such interaction, it's about minus 15 kilocalorie in a mole, and for keta form, and minus 12 for enol form. And also some molecules which are interacting from the edges, from the edge side of nanoparticles, can form covalent bonded complexes with nanoparticles. And you see the geometry of such complex for keta form of thiopurine and for enol form of thiopurine. You see that energy, it's logical that energy of interaction in such covalently bonded complexes is much higher, minus 17, about minus 17 for complex for thiopurine in keta form, and about minus 70, 50. I should say that for a complex of nanoparticles with thiopurine in enol form, it's important that sulfur atom of thiopurine is involved in formation of covalent bonds, which is also important, as I mentioned, for anticancer activity, which is related to existence and the presence of sulfur atom in such drug molecule. For the thioadenine, you can see also the final structures as a result of the modeling, and also we can see that stable non-covalent complexes can be formed with the energy about minus 13 and minus 14 kilocalorie in a mole, and also covalently bonded complexes when the molecules of thioadenine is interaction from the side with the edges of nanoparticles. And also similarly to the situation with thiopurine in enol form, for thioadenine, sulfur atom is involved in formation of covalent bonds. So such interaction can affect the anti-cancer activity of the drug. And the last complexes which we investigated and measured was the nanocomposite of molybdenum disulfide with another fluor-containing derivative of purine nucleobases, 5-fluorouracil. You can see also the peak of PUE, the spectra of PUE drug, and spectra of nanocomposite of molybdenum disulfide with nanocomposite. We are presenting here negative ion mass spectra, and you see that we see the very intensive molecular peak of 5-fluorouracil, and some peaks, cluster peaks, which are related from the nanoparticles, and you can compare this spectrum with the lowest spectrum of poor molybdenum disulfide sample. And based on the spectral data, we did not observe any chemical transformation of fluorouracil within the nanocomposite. Calculation which we also obtained using the same method and the same approach, also provide us with the geometries and energies of interaction in two types of stable complexes which fluorouracil can form with molybdenum disulfide. It can be non-covalent complexes when the plate of the molecules is interaction with the plate of nanoparticles with the energy of interaction about minus 20 kilocalorie in a mole, and the covalently bonded complexes with energy of interaction about minus 47 kilocalorie in a mole. And also it's important that fluor atom is also, fluor and oxygen atom are involved in formation of the covalent bonds. And the conclusion of this investigation are the following. That the structure of anti-cancer drugs and peculiarities of its interaction with nanosheet can influence significantly the behavior of the drugs within the nanocomposite of the nanoplatforms for the drug delivery purposes. As a result of performed LD mass spectrometrical characterization, we can state that nanocomposites of molybdenum disulfide nanoparticles containing anticancer agents doxorubicin and 5-fluorouracil. For this nanocomposite, we did not observe any spectral evidence of the drug's molecule structure transformation, which can be crucial for their therapeutic activity. At the same time, it was revealed the spectrum conformation of some chemical transformations, covalent dimerization and oxidation of sulfur-containing anticancer drugs, in particular thiopurine and thiadenine. We believe that such chemical transformation can reduce their therapeutic activity within the nanocomposite. The results of the theoretical modeling give us the possibility to demonstrate that drugs molecules can form the stable staking complexes within nanosheets surface, and we suppose that these complexes are responsible for effective loading of drugs on this nanoplatform, and also some molecules can form stable covalently bonded complexes with molybdenum disulfide nanosheets. We suppose that data obtained should be taken into account under the development of molybdenum disulfide-based nano-biohybrids for biomedical application as multifunctional drug delivery platform. Thank you for your attention.
Thank you very much. Dear colleagues, thank you very much, Vlada. Very interesting talk, very good combination of theoretical results and computational results. And now we are open our session for the questions. And now I see raised hand from Valina Ivanivna Dolbeshko. Please ask your question.
Vlada, thanks for very interesting report. I want to ask you and also to maybe partially to reply. The first, my question is connected with oxygen again, and you told about that it is necessary to do replacement of sulfur atom. From my point of view, it is not necessary to do because as usually molybdenum S2 has not stoichiometric ratio, and as usually oxygen, not oxygen, sulfur, the amount of sulfur is less than should be according to the formula. That is why you have the sites of localization, nothing localized in these sites. So it is not necessary to replacement. And as why do you use? You agree or not?
We said about replacement in the structure of thiopurine. So this is thiopurine molecules, and this is the hypoxanthine molecule, peak of which we have found in the spectrum. So it's replacement and thiopurine, not in the nanoparticles, Galina Ivanivna. So it's about its explanation why we have seen in the spectrum the peaks of new compounds, which we did not observe in the spectrum of poor thiopurine. So this is replacement related to structure of thiopurine, not in nanoparticles.
Okay, and as to another about application of DOX as one of the example of drug which is necessary for delivery. From my point of view, it is maybe not a proper example because DOX, it is not possible to transformate even under action of electrons. It is formula contain or preserve absolutely the same without changes under electron action, and I think nothing will be under interaction.
Galina Ivanivna, thank you very much. This is discussion, probably some short answer because we
Short answer that molybdenum disulfide is used not by us but in medicine, and I can give you the particles for the DOX transportation, because molybdenum disulfide also providing the photothermal additional therapy of cancer. So it's for synergetic effect for doxorubicin and photothermal therapy.
Okay, thank you very much. Dear colleagues, other